In this video, I want to talk about human and ape genetics. In particular, I want to talk about differences between human and chimpanzee genomes. Now, since the publication of the genomes of humans and chimpanzees, people have been able to do comparisons of the two genomes, and percentage differences have been reported varying from about 2% up to as much as 15%, depending on what is being compared. But the question I want to ask is, are these percentage differences that important, or are functional differences much more important? There are three kinds of functional differences that I want to talk about. The first is orphan genes. Orphan genes are also called taxonomically restricted genes or de novo genes, and they're basically protein coding genes with no detectable homologs in other species. Now, the word orphan derives from the term open reading frame, which is basically a section of DNA that can be read to produce a protein. And these sections of DNA that we find are only in a particular species and not in all species have no mother genes, if you like, so they are orphans. So you take the letters O, R, F and add "and" to them and you get orphan genes. Then there are HARs, which are human accelerated regions of DNA, and these are short genomic regions that are highly conserved across vertebrates in general, but have many substitutions or many differences in humans. And the third interesting feature I want to talk about are HAQERs. HAQERs are human ancestor quickly evolved regions, and according to the evolutionists who have done the analysis, these are the fastest evolving human genomic regions, and they've been identified by the fact that there's extreme mutation density in short windows of DNA. So when were these things discovered? Well, the first reports of human accelerated regions was in 2006, and the first publication of human taxonomically restricted genes, because taxonomically restricted genes have been known for many species for quite a while, but the first human taxonomically restricted genes were reported in 2009. And HAQERs are very recent, first reported in 2022. So let's look at taxonomically restricted genes. These first reported in 2009 in a paper by Knowles et al. And they describe these, what they call de novo genes found in human beings when compared with other species. So to understand what we're talking about here, we need to do a little bit of genetics. We need to think about synteny and taxonomically restricted genes. Here I have a diagram, a very diagrammatic representation of a bit of DNA with the red boxes there, non-coding regions, and these boxes with the pointed ends there are genes, and other bits are non-coding. And we have 4 species here: fish, chicken, chimpanzee, and human. And we can see that here are some genes which are shared by all these 4 species. And these must be very important genes necessary for functioning of living things. Otherwise, they wouldn't be shared across all species. But there are some genes which only appear in the human DNA, here shown by the light blue and the orange genes. These genes are taxonomically restricted, which makes them different from the synthetic genes which are shared across all species. Evolutionists believe that they came about by the process of mutation, which created new open reading frames and promoters and transcript structures. This is believed to have occurred by the process of gene duplication plus rapid sequence change, and possibly by the recruitment of transposon-derived sequences into new gene structures. They're very important because they have roles in brain development, reproduction, and immunity, and they are involved in various regulatory processes, and when these processes are disturbed, cancer seems to result. Now, there are hundreds of these genes, so how did these genes come about? The origin of these taxonomically restricted genes is a bit of a puzzle. Everyone agrees that it's very implausible, yet evolutionists have to think that it is possible because they actually exist. So evolutionists have to use their imagination to explain their origin. And a good example of this is a paper written by Weissman in 2022 attempting to explain the origin of these de novo genes and to overcome what's known as the apparent implausibility of de novo genes. Now, Weissman proposes various mechanisms along with her colleagues, and these mechanisms are actually admitted to be very speculative, and the suggested causes are accepted to be highly conjectural. And despite the fact that these mechanisms which are proposed are basically imagination, evolutionists are kind of constrained, forced to accept that evolution can produce these genes because there's no other acceptable explanation for an evolutionist. Right now, let's look at human accelerated regions. Human accelerated regions were first published in a paper written by Pollard and his colleagues in 2009 when they did some comparative genomics of the human and the chimpanzee genome, and the analysis revealed 202 segments, which they said have extensively changed in the last 5 million years. So let's have a look at our diagram of the genome again. And here we see there are highly conserved regions in other vertebrates. And we're not actually looking at genes in this case, we're looking at the boxes, which are not genes. These are regulatory regions, not regions which produce genes. And we see that in the chicken, chimp, and human, they are highly conserved. We've got the purple boxes here, but in the human regulatory region, there are changes. And we've got three of these illustrated in this diagram. So these sections of DNA are not used to make proteins, but they can be thought of as switches which actually regulate the functioning of genes. So what about their origin and function? Well, again, according to evolutionists, these genes came about by many nucleotide substitutions, as many as 25 mutations in one HAR in the human genome, and this all happened since the divergence from the common ancestor with chimpanzees. And they believe that this is adaptive evolution acting on random mutations. And multiple HARs function as enhancers in neural and brain development. So they're very important functional regions. But according to evolutionists, this happened even though it is highly improbable. They have to believe that even though one beneficial mutation is unlikely, as many as 25 mutations in any one HAR have occurred, and all of these must confer selective advantage, or at least neutral, to become fixed in the ancestral human population. Now, there's a big problem for the origin of these HARs because they are in areas which are highly conserved across different species. So there's what's called strong purifying selection in the evolutionist world. And this basically means that these regions have been conserved because every alternative is harmful to the organism which carries them. So that's why there is what's called strong purifying selection, which maintains the sequence. But then suddenly in the human lineage, we find that they've changed. Now, we know they're important, and we know that this is highly unlikely because any perturbation of the regions can alter gene expression. And when this happens in the human genome, we have neurodevelopmental problems as an example. So this highlights the fact that strong purifying selection has kept these regulatory regions the same in every species except in humans. So we'll come back to that later. So let's think about human ancestor quickly evolved regions. These were first reported in 2022 by Mangan and his colleagues in a paper called Adaptive Sequence Divergence Forged New Neurodevelopmental Enhancers in Humans. These basically are claimed to be the fastest evolving regions in the human genome. And latest analyses have detected about 3,000 of these HAQERs. Again, this is done by comparison of the genomes of chimp and humans. So let's think about the origin and function of these human ancestor quickly evolved regions. They have been identified again by comparative genomics and counting unusually large numbers of differences in short sections of DNA. So evolutionists say that this is because of extremely fast mutation rates. Now we have as many as 29 mutations in any one HAQER. Evolutionists talk about short bursts of intense positive selection. One wonders how they know that that happened. But these genes, again, are regulatory regions which apparently seem to be poised for activation during development, especially in the brain and the gastrointestinal tract. So they are very important. They're important from the point of view of controlling genes. They're regulatory sections. They act as enhancers, switch-like elements, influencing gene expression, and in particular in the development of the cerebral cortex, the part of our brain responsible for higher functions such as language and thought. Okay, but I would like to suggest to you that these features are all evolutionarily impossible. They are very improbable, and basically, when you look at the probabilities, you come up with a problem because you need too many specific mutations. The problem with these is it takes time to fix a new mutation into the genome. And this problem is especially significant for humans because the generation time is quite long, 20 years. And also there's a problem of population size. Now, evolutionists say that the ancestral population of humans was around 10,000 individuals at most. So let's look at the problem for HARs, for example. HARs are in areas which are highly conserved across vertebrates. And this generates a problem for evolutionary explanations of HARs if they are highly conserved due to purifying selection. It's very difficult to see how changes could be made which are not fatal to the organism carrying them. All alternatives basically are harmful, and HARs are regions that typically have 10 to 25 differences when compared with other vertebrates. Now, the probability of getting one mutation is about 10 to the minus 8. The probability of getting, say, 10 is 10 to the minus 80. And this is basically impossible. There are less than 10 to the 80 atoms in the universe, so you haven't got a possibility of generating anything that has a probability of 1 in 10 to the 80. Even if 10 specific mutations are highly improbable, 25 are even less probable, the whole issue becomes impossible. So there must be a better explanation, and as a creationist, I believe there are better explanations. Orphan genes, HARs and HAQERs, have the hallmarks of design because of their important function. They are specific genes in the case of orphan genes, or they are very specific and important control regions in the case of HARs and HAQERs. They're designed to give us, among other things, big brains. And the only reasonable explanation is that humans had them from day one. The probability of evolving them in the timescales that evolutionists propose is essentially zero. Just 4 features which highlight some of the differences between apes and humans. Just to finish, I want to look at the thumb, the chin, the leg, and the brain. We have opposable thumbs. If I hold up my thumb, you can see I can bring my fingers onto my thumb without any problem. Now, apes can't do this. The opposable thumb is a feature of humans. We have chins. We all have chins. Apes do not have chins. We have legs which are designed for standing, walking and running, and even doing ballet. A chimpanzee can hardly stand and can certainly not walk very far before having to drop onto all fours. And then we have our big brains for language and rational thought. And these features of the human being are controlled by our genomes. And these features that we've looked at, these 3 features of the human genome, orphan genes, HARs and HAQERs, are part of the control features that allow human beings to exist. And as a creationist, I see these as design features and illustrating once again how fearfully and wonderfully we have been made by our Creator God.